3 billion base pairs. That's how much DNA sits coiled inside almost every cell in your body right now. Fewer than 2% of those 3 billion letters ever get translated into a protein. The other 98% was formally classified in published scientific literature as junk, a specific term coined by a geneticist in 1972, taught in textbooks as settled fact for 3 decades afterward. In October of 2024, 2 scientists won the Nobel Prize in Physiology or Medicine for taking a piece of that exact junk seriously enough to find a working signal buried inside it, one now known to help run a majority of the genes everyone had already agreed were the real, legitimate ones. Here's the belief I want to take apart, because almost everyone watching this carries some version of it without ever stating it out loud. The idea that a stretch of DNA only matters if it eventually becomes a protein. Proteins build the physical structures in your cells. Proteins carry out the chemistry. Proteins are the working molecules. So DNA that never produces one gets treated as leftover material from millions of years of evolutionary accumulation, present but inert. It's a reasonable belief. It matches what most people are taught about gene expression. DNA gets copied into RNA. RNA gets translated into protein. Protein does the work. That's a real process, and it's accurate as far as it goes. The problem is that this belief, treated as the complete picture, was already obsolete by 1993. And almost nobody outside one small research group in New Hampshire noticed at the time. The lab belonged to Victor Ambrose, and the organism he studied was a millimeter-long roundworm called Caenorhabditis elegans, a creature that carries roughly 19,000 protein-coding genes of its own, a number close enough to the 20,000 or so found in the completed human genome that it quietly embarrassed a lot of people who'd assumed human complexity had to live in a longer gene list. These worms are also unusually precise animals. Every adult hermaphrodite grows into exactly 959 somatic cells. Not approximately, not on average, exactly 959. And the full lineage of every one of those cells, traced back to the fertilized egg, had already been mapped division by division. That precision is what made one particular mutant so strange. In a normal worm, a gene called Lin14 is highly active early in development and then gets shut off on a fixed schedule, so the worm's cells stop behaving like larval cells and start behaving like adult cells at the right time. In certain mutant worms, Lin14 never received that signal. It stayed, switched on, and the worm kept repeating its larval stage development well into adulthood. Something was clearly responsible for silencing Lin14 on schedule in normal worms. And whatever that something was, nobody had found a protein that did it. The gene responsible turned out to be called Lin4, and when Ambrose's group finally isolated it, they found something that didn't fit the standard model of gene regulation at all. Lin4 didn't code for a protein. There was no functional stretch for building an amino acid chain, nothing for a ribosome to translate into a working molecule. What Lin4 produced was a short strand of RNA, about 22 nucleotides long, that never advanced beyond being RNA. And that short strand worked in a direct, almost blunt way. It base paired with a matching sequence sitting in the tail end of the Lin14 messenger RNA and physically attached to it. The amount of Lin14 messenger RNA in the cell barely changed. The amount of Lin14 protein the cell could produce collapsed, because a ribosome cannot cleanly translate a messenger that has another strand of RNA bound to it. 2 research groups published this discovery in the same year, 1993, in the same journal. Ambrose's team and a second team led by Gary Ruvkun, who had been tracking the same regulatory puzzle from the opposite direction. Between the 2 papers, they had identified a new category of gene control entirely, an RNA molecule that never becomes a protein, regulating a gene by binding directly to another RNA molecule, with no protein intermediary at any step. Consider what that actually requires you to revise. The standard model assumed regulation happened upstream at the DNA itself, through proteins called transcription factors attaching to specific control sequences before a gene was even copied into RNA. Lin4 bypassed that whole stage. It regulated its target after the messenger RNA already existed, using a short fragment of RNA to interfere with a different RNA directly. This wasn't a minor variation on a known mechanism. It was a distinct regulatory system operating in parallel with the one already described in every relevant textbook, using the same chemical class of molecule the gene's own output was made of. For contrast, look at what happened to a related discovery made 5 years after Lin4 in the same worm. In 1998, Andrew Fire and Craig Mello showed that double-stranded RNA injected into Caenorhabditis elegans could silence a specific gene by triggering destruction of its messenger RNA, a mechanism they named RNA interference. It's mechanistically distinct from what Lin4 does, but it belongs to the same broad category, RNA controlling gene expression directly, with no protein doing the controlling. RNA interference got recognized fast. Fire and Mello won the Nobel Prize in 2006, 8 years after their paper, largely because the technique was immediately useful. Any laboratory could use it to switch off a chosen gene on demand and observe what broke. Lin4 offered no equivalent shortcut. It didn't hand anyone a tool. It only described how one organism actually regulates itself. And in a field oriented around utility, a description with no obvious application apparently generates a very different kind of interest than a technique does. The kind that took 31 years to convert into formal recognition instead of 8. You'd expect a finding like that to reshape the field immediately. It didn't. For most of the following decade, Lin4 sat in the scientific literature as a curiosity, cited occasionally, taught rarely, and treated by a large share of working molecular biologists as a nematode-specific mechanism, something worms had developed for their own narrow developmental timing, and nothing more general than that. That reaction deserves attention, because it's the exact same belief I opened with, just held by trained specialists instead of casual observers. If a regulatory mechanism doesn't resemble the mechanism you already trust, if it's RNA instead of protein, produced by a gene with no coding sequence, the instinct, even inside the profession, was to file it as an exception rather than a principle worth generalizing. Ambrose experienced the cost of that skepticism directly. Around the period this work was completed and published, he was denied tenure at Harvard. He rebuilt his research program at Dartmouth Medical School and stayed there for roughly 15 years before eventually moving to the University of Massachusetts. That's not a sympathetic aside about a difficult career. It's a data point on how seriously the field weighed a discovery that the Nobel Committee would later describe as revealing a completely new principle of gene regulation. The finding was correct in 1993. It simply didn't resemble anything the field was prepared to recognize as significant. What eventually forced a reconsideration wasn't a better argument for the same result. It was a second example. Because in biology, a second example is usually what does the actual work of proof. It lets you ask the one question that separates a coincidence from a principle. Does this show up anywhere else? In 2000, Ruvkan's lab identified a second small regulatory RNA in the same worm, called LET7. A second worm gene, on its own, wouldn't have moved the field much. What mattered was where else they found it. When they checked the same sequence against other animals, LET7 turned up nearly unchanged in flies, in mollusks, in fish, and in humans. A stretch of regulatory RNA conserved across roughly 600 million years of separate evolutionary history, the kind of conservation biologists typically reserve for genes so fundamental that losing them is lethal. That result did what the 1993 papers alone hadn't managed. It told the field, without much room for alternative interpretation, that Lin4 hadn't been a private trick of one species. It was the first sample of something universal, discovered first simply because worms were the easiest place to find it. Before I go further with the scale of this, I want to flag something I've gotten wrong before, because there's a companion claim to this story that circulates constantly and doesn't hold up as cleanly as the microRNA discovery does. In 2012, the ENCODE project, a large international genomics consortium, announced that roughly 80% of the human genome shows some form of measurable biochemical activity, and several of its authors argued this meant the term junk DNA should be retired outright. That figure got repeated everywhere, including in earlier explanations I've given, as though it settled the question. It doesn't. A group of evolutionary biologists, most prominently Dan Grauer and several co-authors, published a detailed rebuttal arguing that biochemical activity and evolutionary function are not interchangeable measurements, that plenty of DNA can bind a protein or get copied into RNA without that activity affecting the organism's survival at all. Under the standard evolutionary test, checking whether a sequence shows the signature of natural selection actively protecting it from mutation, independent estimates put the functional fraction of the human genome somewhere between roughly 5 and 15%, not 80. That disagreement is not resolved. It's an active, unsettled argument in the field, not a rhetorical flourish about science, always questioning itself. So the honest claim isn't that essentially all of your genome turned out to matter. The honest claim is narrower, contested, and considerably less tidy than the ENCODE headline suggested. What does hold up, independent of that fight entirely, is that a specific, well-defined slice of the non-coding genome produces molecules that actively and measurably control which genes get expressed. That claim doesn't need any inflating. It's strange enough as it stands. Back to scale, then using numbers that do hold up. In 2001, 3 separate research groups. Ambrose's lab, a lab led by David Bartel using new techniques built specifically to isolate tiny RNA molecules from cells, and a lab led by Thomas Tuschl, published papers in the same issue of the journal Science. Together they reported dozens of new microRNAs in worms, flies, and humans in a single publication cycle. From there, the pace accelerated. Current catalogs list well over 1,000 distinct microRNA genes encoded in the human genome, which is the figure the Nobel Committee itself cited in 2024, with more exhaustive databases listing upward of 2,000 candidate sequences, though only a portion of those meet the strictest confirmation standards. Here's the number that should actually reorganize how you think about the 2% of your genome you were taught to respect. MicroRNAs are estimated to regulate more than 60% of all human protein-coding genes, and a single microRNA can suppress production of hundreds of different target proteins at once, because the recognition rule turned out to be far looser than Lin4 and Lin14 first suggested. A microRNA typically only needs a short stretch of 6 to 8 matching letters, not a full-length match, to grab hold of a messenger RNA and interfere with it. Loosen the matching requirement, that far in one short RNA molecule stops being a specialist aimed at a single target and becomes something closer to a dial that turns down an entire category of proteins at once. That's not 60% of the genome. That's 60% of the genes everyone had already agreed were the legitimate, functional, protein-producing part. Think through what that actually implies. The portion of your genome universally agreed to matter, the protein-coding 2%, is extensively supervised by molecules manufactured out of the portion universally agreed not to matter. This isn't a case of the 98% quietly running its own minor operation off to the side. A meaningful fraction of it sits upstream of the 2%, determining which of those genes actually get expressed, in which cell type, at what level, and on what schedule. The assumption most people still carry, genes at the top of the hierarchy, everything else beneath, is backwards for a substantial share of the genome. Disrupted microRNA activity is now directly linked to cancers, heart disease, and viral infection, which is exactly the kind of medically serious territory the inert filler framing would have written off by definition before anyone checked. None of this stayed confined to laboratory papers, and the way it eventually got formally recognized closes the loop on where this story started. In October of 2024, 31 years after the original Lin4 papers, the Nobel Assembly in Stockholm awarded the prize in physiology or medicine jointly to Victor Ambrose and Gary Ruvkun for discovering microRNA and its role in regulating gene expression after a messenger RNA has already been produced. The committee's own description called it a completely new principle of gene regulation, essential for how complex, multicellular organisms, including humans, actually develop. Ambrose received the honor as a professor at UMass Chan Medical School, in the career he built after Harvard denied him tenure during the exact period this research was underway. Ruvkhan received it at Harvard, where he had remained the entire time. Between the two of them, a finding that spent its first decade being treated as a footnote about worm development became one of the most significant recognized principles in modern molecular biology, sitting the whole time inside the part of the genome that already had a dismissive label attached to it before anyone had properly checked. There's a reason this extends past a footnote about gene regulation. Every cell in your body, a neuron, a liver cell, a heart muscle cell, carries the identical 3 billion base pairs you started with as a single fertilized egg. The protein coding instructions are the same file, copied into every single cell without exception. What makes a neuron function like a neuron instead of a liver cell isn't a different set of genes. It's which genes are permitted to be expressed, at what level, on what schedule. And a substantial share of that decision is executed by microRNAs, manufactured from the layer of the genome that spent 30 years labeled as waste. The part of your DNA once classified as junk isn't a footnote to what makes you a specific, differentiated collection of cells rather than 3 billion identical instructions expressed identically everywhere. In a direct, mechanistic sense, it's closer to the layer responsible for that difference than the protein-coding genome itself is. So when someone repeats the claim that most of your DNA doesn't do anything, the accurate response isn't a vague objection that biology is more complicated than that. It's a specific, testable one. That exact claim was investigated, it failed, and it failed because doing something was never actually the same question as coding for a protein. Ambrose and Ruvkhan didn't discover that junk DNA secretly makes proteins after all. They discovered that the entire assumption underneath the junk DNA label, that gene regulation requires a protein, was the actual error, sitting undetected for 3 decades because it had never been tested as a hypothesis. It had been treated as a definition. Junk DNA was never a fact about your genome. It was a confession about what we were willing to count as a signal.